Hello, everybody. I am Kuwolee Sharma. I am a first year student at St. Joseph's University, Bangalore, and I'm pursuing my bachelor's degree in biochemistry and zoology. So, today I'm going to speak about the origin and conduction of nerve impulse. So, before we get into the technicalities, let us first see what is a nerve impulse and why is it so important that we read it as a separate topic. So, nerve impulses are also known as action potentials, which are basically electrical signals generated and transmitted by neurons, the specialized cells of the nervous system. These impulses play a crucial role in communication within the body, allowing for rapid transmission of information between different parts of the nervous system and coordinating various physiological processes. But why are these so important? So, nerve impulses are fundamental to the functioning of the nervous system and are integral to numerous biological processes, from basic reflexes to complex cognitive functions, highlighting their critical importance in maintaining overall health and functionality. So, before we delve into nerve impulses further, let us first see what is the specialized cell of the nervous system. So the specialized cell of the nervous system is the neuron. So this is the neuron. I won't go deeper into the structure of the neuron. All we have to care about is the dendrites, the cell body, the axon, and the axonal terminals. Why only four of these are very important is that the dendrites of the neuron receive the information, the cell body processes and integrates that information, the axon carries the information along all the long distances, and the axon terminal transmits the information to the next cell in the chain. A bundle of these axons together are called a nerve. A nerve can be very long, and we won't go in that direction. And how the impulse is transferred from the dendrite to the axon terminal, this is the direction of the nerve impulse. This is how the impulse is taken throughout. So let us see what happens within a nerve impulse. So let us first see the resting membrane potential. So what is the resting membrane potential? Resting membrane potential refers to the electrical potential difference across the membrane of a neuron when it is not actively transmitting a nerve impulse. So this is the potential of the neuronal membrane before a stimulus acts on it. So this potential difference is maintained by the distribution of ions or charged particles inside and outside the neuron. So even the neuron has a lipid bilayer like a plasma membrane, which is known as the exolemma or the neurilemma. So at this stage, the neuron's membrane is selectively permeable, meaning it allows only a certain category of ions to pass through it while restricting others. The primary ions that are involved in establishing the resting membrane potential are sodium, potassium, and chloride. So what is the ion distribution inside and outside the lipid bilayer? So the ion distribution is as such that the potassium ions have a higher concentration inside the membrane and a lower concentration in the extracellular fluid. And the sodium ions have a higher concentration outside the membrane and a lower concentration in the extracellular fluid. The chloride ions have moderate concentrations inside and outside the extracellular fluid. So what basically happens in the resting membrane potential is that there is an efflux of potassium ions and an influx of sodium ions, which creates an ionic difference, which is known as the membrane potential or the electrochemical gradient, which is equal to minus 70 millivolts. Moving further, we have, so this is a visual representation of the resting membrane potential of what basically happens. The potassium ions move out and the sodium ions move in. These diamond-shaped structures are the potassium ions and the round structures are the sodium ions. So next we have the depolarization phase. So what does depolarization mean? Depolarization refers to the phase of action potential or the nerve impulse where the membrane of the potential where the membrane potential of a neuron becomes less negative or even it becomes positive because of a stimulus of 15 to 20 millivolts, which is also known as a graded potential. So when a stimulus is received by the neuron, it triggers the opening of voltage-gated sodium channels in the neuron's membranes, and the potassium channels close. Because of this, there is an influx of sodium ions which causes the membrane potential to become less negative and in cases even positive. This shift of membrane potential from minus 70 millivolts to minus 55 millivolts to almost plus 30 to plus 40 millivolts is known as depolarization or depolarization phase. During this phase, the membrane is 1,000 times more permeable. The depolarization phase is crucial for initiating and propagating nerve impulses along the axon of a neuron. Next, we have repolarization phase. So repolarization phase is the phase of the action potential or the nerve impulse where the neuron's membrane potential returns to negative resting phase after depolarization. This involves the efflux of positively charged ions, primarily potassium ions, out of the neuron. Potassium ions move out of the neuron into the extracellular fluid driven by the concentration gradient and the electrical gradient. As potassium ions leave the neuron, the membrane potential becomes more negative and it gradually returns to the resting membrane potential, and the potential approximately becomes minus 70 millivolts. At this stage, there are three things happening. The sodium channels open, the potassium channels open. Also, the sodium potassium ATPase pumps come into action, which which is basically an active process. So this entire process is called repolarization. It is essential for restoring the neuron's membrane potential to its resting state, allowing the neuron to generate subsequent action potentials and maintain its responsiveness. So this is a visual representation of depolarization and repolarization. So what happens is in depolarization, the sodium, only the sodium channels are open and the potassium channels are closed. We can see that only the sodium ions are moving out. The potassium ions are not moving anywhere. Whereas in the case of repolarization, we see that the potassium channels are open. Also, the dumbbell-shaped sodium-potassium ATPase pumps are in action, which bring in 3 sodium ions and 2 potassium ions in and out of the membrane. The last and final stage is called the hyperpolarization phase, which refers to a state where the membrane potential of the neuron becomes more negative. Like there are chances where the membrane potential goes down from minus 70 to almost minus 90 millivolts. This means that the inside of the neuron becomes more negative compared to the outside. So Why does this happen? One of the main reasons why hyperpolarization occurs is the continued or efflux or the outward movement of potassium ions from the neuron. After an action potential or the nerve impulse, the voltage gated potassium channels remain open for a short period, allowing more potassium ions to leave the neuron than usual. So basically, the potassium channels after a nerve impulse take a bit longer time to close. They are not very fast in the case of closing the channels. This is the graph for the electrical activity that we have seen so far, which explains how the action potential moves, how it goes to the. So we can see this portion is the resting membrane potential, then the stimulus is received, then it goes into depolarization phase. This peak is known as the action potential. Then it comes back towards resting potential, which is known as repolarization. Sometimes or mostly it becomes more negative, which is the hyperpolarization state, and it comes back to resting phase, that is minus 70 millivolts. So that was it. I hope I was able to explain the topic very well. Thank you.